Doenças Infecciosas e Combate a Agentes Patogénicos, e tenho o prazer de apresentar um painel de oradores absolutamente excelente. Nós vamos fazer a sessão em inglês, foi o que combinamos, a não ser que haja aqui alguém que por acaso não perceba inglês ou que se oponha a que seja em inglês. Não? Se preferir falar em português, pode falar em português. Ok. Pronto, tudo bem. Ok. So to start, we are going to have a change in the program. And the first speaker will be Margarida Saraiva, from Instituto de Investigação e Inovação em Saúde, o ITRES do Porto. Then she will talk about the new knowledge and pathogenicity of tuberculosis.
Okay, so good afternoon, everyone, and thank you very much for this invitation. My slides are actually in Portuguese because I wasn't sure about the language, so I'll speak in English, but the slides are in Portuguese. And I'm going to talk about lung infectious disease. So what a year to talk about lung infections. And the infection we study in my lab is tuberculosis. This is our institute, for those of you who have not been there or haven't seen it. I don't know if there is any. Okay, so this is I3S. It's not always as sunny in Porto, as Maria João knows, but this was a nice day. This is our lab, and we are supported by all the scientific platforms that we use to work with a pathogen that actually infects and kills many people every year. So tuberculosis is classically the big killer in terms of infectious diseases. In the last 200 years, tuberculosis claimed about 1 billion lives, so much more than all the other considered big killers like smallpox, 300 million, malaria, influenza, cholera, AIDS. And the deaths by COVID-19 are about 6.7 million at the moment, and hopefully will decelerate because of all the vaccination efforts. So tuberculosis is also a disease that is very much portrayed in many different artistic like opera, paintings, books, etc. But unfortunately, it is not a disease of the past. So tuberculosis remains the first cause of death by a single pathogen, a single bacteria, until very recently by a single pathogen. It is really spread through the entire world, but it is much more or cause of concern in certain areas, as you can appreciate from this graph, where the incidence is very high. So there are about 1.4 million deaths of tuberculosis per year and 10.4 million new cases. And the scenario is not very optimistic considering the 500,000 cases that are resistant to antibiotherapy and that are increasing over time. So another concern related to TB, to tuberculosis, is that it really perpetuates poverty, and poverty perpetuates TB. So it's a very difficult cycle to break. And so it is not surprising that ending TB is within the United Nations Sustainable Development Goals. So with the aim of eliminating TB, so that's the UN Sustainable Goal number 3, or within the Sustainable Goal number 3, what do we need? What do we have to do? And basically, we need to act in different fronts. We really need new diagnostics because the ones that we have take a long time. So by the time the person is diagnosed with TB, it has spread the disease to others. We also need new therapies because, as I said, resistance to biotherapy is increasing. And importantly, every time a new antibiotic is introduced, the resistance to that antibiotic comes in a shorter period than to the one before. So this is a very fast-moving field. So we need new therapies. And we also need new vaccines because the one that we have, and that is over a century old, that's BCG, does not protect completely to the disease, and specifically in terms of human pulmonary tuberculosis, is not a very efficient disease. And then we also need a better monitoring of the cases, and this is improving a lot with whole genome sequencing techniques. So in my lab, we are not dedicated to any specific, those specific points as a single or unique aim, but we think that the best way to contribute to this is to understand better the pathogen, our enemy, which is Mycobacterium tuberculosis. And you can see here the way a colony of Mycobacterium tuberculosis in agar looks like. It's actually very pretty. And here you can see different bacteria, so in green, infecting a macrophage, a host macrophage. So this is our aim, to understand our enemy. But we want to do it in a particular way. We want to understand it in its natural habitat, if you wish. So our vision is that we know now that the bacteria that cause tuberculosis have evolved with the human population and have accompanied the expansion out of Africa movement. And with that, the bacteria and the human populations have really found a system to co-evolve, and they are now phylogeographically related. So this, in simple terms, means that we don't have a single type of bacteria, but instead we have very different human-adapted bacteria that cause tuberculosis in different places. So our strategy has been to study how these different bacteria interact with our immune system, looking from clinical isolates and of different locations. So we have studied lineage 2, that's the lineage that exists in East Asia. We have also published studies in bacteria from lineage 4, and I'll show you a little flavor of this. So this is the lineage that is more prevalent in Europe and in Portugal. And we have also started some studies in lineages 5 and 6 that are restricted to West Africa. So this is also a very interesting study, but I'm not going to talk about it. So what have we done in this study that you can then read in more detail? So we thought if we want to look at the way the bacteria interact with the immune system, and if we want to look at it from a more natural perspective, we need to start from where the bacteria exist and where they are causing problems, and that are the human patients. So we analyzed clinical data from a cohort of over 600 patients in Porto, and each patient was basically assessed to obtain a scale of TB severity. So we had mild disease, moderate disease, and severe disease. And once we saw this, what we did was to now grow the bacteria in each of these groups. So these are a lot of isolates that we grew at the time. And we performed some bacterial genetics experiments on, or analysis, better saying, on these bacteria. And we were able to define that our bacteria were basically almost all belonging to lineage 4, so the European lineage as expected, with only a few exceptions. But interestingly, and although they were all from the same lineage, may I remind you that they did cause diseases of different severities. And when we looked at the whole genome, we started seeing some hotspots that could perhaps be responsible for these differences. And moreover, we pursued then a series of in vitro, and now more recently, so not published as yet, in vivo experiments, where we combine infection of human cells with the different isolates that we extracted from these patients here. We measured cytokine responses, we performed whole transcriptome analysis, and then we did some animal experiments using the mouse model of aerosol infection. And with this combination of techniques and methods, we figured out a novel, or we proposed a novel mechanism of immune evasion that is developed or evolved by Mycobacterium tuberculosis to really escape the surveillance systems that exist in our macrophages, and in these particular isolates that manage to escape, they really tune the immune response to a very low level. And this is then reflected in a more severe disease. So this is one of the first studies really using Mycobacterium tuberculosis diversity as a tool to understand new mechanisms of immune evasion and how they articulate disease severity. And what is really fascinating for us, at least, is that we can see this mechanism across different human donors and even across different species, because we see it in the human and in the mouse immune system. So it's a very powerful mechanism developed by the bacteria. So why is this important? To go back to my initial slide, how can this help in developing new tools to fight TB? So as I told you, antimicrobial resistance is a problem in tuberculosis. We need new therapies, and many people are developing therapies based in the modulation of the immune response. So these are therapies that basically are focused in the human immune system, so they are not prone to develop resistance developing because they do not affect directly the bacteria. And the idea is to modulate the immune system to the right level to cause the minimum disease. Now, if we have a different bacteria that modulate this immune response, we must incorporate that dimension of the diversity of the pathogen so that we can develop the best therapy. Otherwise, we modulate the immune system to be here, and then our bacteria goes up or down. So we need to really consider these host-pathogen interactions for the development of immune-based therapies. So just to finish, in a more philosophical note, ending TB is really our aim. And this is like the tip of the iceberg. And to get to here, we need new vaccines, new therapies, and we need things that people can see, and then we can, it's tangible, we can feel the impact of this. Where we work is under the water. So we work on the massive amount of fundamental science that is needed to feed into this. And because this is a meeting where the general public is also present, I always like to show this slide that what society sees and wants is really solutions here. And what we are doing is to work towards those solutions with a lot of fundamental research. So investment from society will come back to society. And I think, of course, I normally show this for tuberculosis, but you will all agree with me that with the COVID-19 situation, this is perfectly illustrated as well. So it's a very nice cycle that we have here. So I'll just finish by thanking the people who did the work, so that those are the people in my group who work every day very hard to this, the I3S scientific platforms that helped us, and then a series of I3S collaborators, national and international as well, and the funding agencies that have supported the work over the years. So I also thank you for your attention.
Do we have questions from the audience?
Thank you. Should we consider TB as a zoonose that should be addressed in a One Health approach?
In my personal perspective, it's not as much as a One Health situation like classically, because it is very human adapted. What is something that I sometimes propose, and it's a bit unconventional, is that in most situations, Mycobacterium tuberculosis is like a semi-commensal. So it doesn't cause disease in every single person that infects. There are many people that survive with latent infection. And I think that would be, to me, a nice perspective to tackle the question, because It isn't all about disease, if you see what I mean. So it's a global problem, but it's more to it than the active disease with the symptoms and the contagion going on.
More questions? If I may. You mentioned, and you have seen in the map, that one of the most affected areas are areas that are poor areas by excellence. And you said that very well. And I was wondering whether it's not just developing faster diagnoses, they also have to be cheaper and easy to use. So what is the situation on the development of these diagnostic therapies, if you know anything about this, and how can your program also be applicable within these poorer countries?
So I think the main problem in these specific areas is related, on one hand, it's true with diagnosis because there is some laboratory equipment that is required, but it's not the most difficult thing to do. We still do Zell-Nielsen staining of sputum, so it's still in TB everything is very old. It seems that a lot of research is being done, but the techniques that are used are always the same as they were years and years ago. I think the main problem, to answer your question, in these countries is the treatment. The treatment to TB is very long, it's 6 to 9 months, and it's very toxic. And after the first 2 weeks, people start feeling better. Because from the WHO guidelines, you have to be, the treatment is under direct observation, so if you have tuberculosis, you don't take the pills home to have them, you have to go every day to the center and to take the pills in front of someone responsible. So basically, many people just abandon the treatment because they have to walk a lot to go to the center, they are feeling better, they want to go to work, and this also happens, for example, in Portugal to some extent. So that gives space for antimicrobial resistance and also for relapses of TB. So the person is feeling better, but it's not cured, it may still be contagious, it's infecting other people, and the problem is just spreading more and more. So I think that, to me, is the main problem hampering TB control.
Thank you so much, Margarida, and thank you for being here. Is there any other question?
That problem that the person can't have the pills because think he looks well or that. The problem will be not solved, but will be better if the formulation of the drugs was be like the pills, for example, for transdermal. Do you think it will
I think what we need most of all is less toxic and less lengthy. So we need shorter regimens to treat TB. If we get to shorter regimens and less toxic, I think that will be a major step. And one of the possibilities is to combine, not to totally abandon antibiotics, but to combine that with novel strategies, namely by modulating the immune response, because we know a lot about immune response that is beneficial or detrimental in tuberculosis. So if we can combine the antibiotic with the help of the immune system, I think we will manage the main problem hampering TB elimination. To me, personally, that's what we work towards, to be fair.
Thank you for the sake of time.
Thank you very much.
Thank you very much. So our second speaker is Nuno Taveira from Centro de Investigação, interdisciplinar Egas Moniz, and we will be talking about the development and evaluation of medicines for the treatment and prevention of infection by HIV. I welcome you. The floor is yours.
Okay, thank you very much. So I will tell you about our small efforts to try to produce better medicines for HIV. You know that HIV is still a major problem. It's still killing a lot of people every year. So far, almost 33 million individuals have died from this disease. That's 8 times more than the COVID problem. And in 2019, still 38 million individuals were living with HIV, and more than 500,000 died of this disease. The major problem here is that of all people living with this virus, only 81% know their status, 61% assessed treatment, and of these only 59% were suppressed, so were not transmitting the virus. The situation has gone better since the early years, since 1990s or early 2000. And as you can see in these curves, both the incidence, the prevalence, and the number of deaths, both the incident cases and the deaths are decreasing sharply, but this is due to the antiretroviral drugs. But of course, people live longer, so the prevalence cases are increasing. And this is especially true in the sub-Saharan Africa. Of course, the presence of HIV is making these countries poor, and we need to tackle this problem. And fortunately, there are many antiretroviral drugs available. They are shown here that prevent all steps of the virus life cycle. But there is a problem of long-term safety, a problem of tolerability. Some of these drugs are unable to treat the immune dysfunction induced by this virus, and especially the major problem is the drug resistance, which is emerging, and some countries report almost 30% of infections with drug-resistant viruses. So another problem is that there is not only HIV-1, there is also HIV-2, and within HIV-1 there is HIV-1 group O, and both HIV group O and HIV-2 are resistant to some of the drugs, namely the non-nucleoside reverse transcriptase inhibitors and fusion inhibitors. And in the case of HIV-2, only some of the protease inhibitors are active against this virus. Furthermore, HIV-2 develops very rapidly resistance to all the available drugs. And in one recent study performed in Cape Verde, we found that almost 60% of the individuals were infected with drug-resistant viruses. So we need new antiretroviral drugs that work better against these resistant strains. And we have devoted some time to design, develop, and test one peptide that was intended to prevent the fusion of the virus and entry of the virus into the cells. We used as an inspiration this enfuvirtide, this T20, that is a peptide derived from this helical region 2 in the GP41 transmembrane glycoprotein, and this peptide binds to the helical region 1 and prevents the structural changes that are needed for fusion to occur. We decided to produce a new and a better peptide because this enfuvirtide does not work on HIV-2, and so we produced ancestral sequences, ancestral transmembrane sequences based on recent sequences from HIV-2 and SIV, and we produced some peptides that are shown here and tested their activity against HIV-2 and against HIV-1. And of these three, P1, P2, and P3, only P3 was active. So I'm going to tell you a little more about this peptide. So this peptide works very well against both HIV-1 and HIV-2, especially against HIV-2, but also very well against HIV-1. It works very well against drug-resistant strains of HIV-1, namely HIV-1 that is resistant to the enfuvirtide, to T20, and it's a fusion inhibitor as shown by time of addition assays. And we then looked further with a collaboration with a Chinese researcher. They produced some modified versions of this P3 with a methionine and a threonine in the beginning of the peptide. This increases the binding of the peptides to the target, and it was found that these modified peptides, some of them had even more potent action compared with P3, and the best ones were these HP23 and 2P23, and they worked well against HIV-1, HIV-2, and also SIV. And of course, then we tested the activity in humanized mice model, and we found that the best of these peptides was 2P23 that works, impedes the infection of this humanized mice model very well. More recently, we found that people infected with HIV, HIV-1 and HIV-2 produce antibodies against P3 and also against G20. The level of antibodies varies with both peptides. People infected with HIV-2 produce more antibodies against P3, and people infected with HIV-1 more antibodies against T20. We decided to look at the neutralizing activity of these peptides. And so we inoculated some mice with these peptides free or in nanoparticles. They produced high levels of antibodies, and in this set of animals, they were neutralizing. So they induced neutralizing antibodies against both HIV-1 and HIV-2. So of course, this is very interesting because this defines a new neutralizing epitope in HIV. And it's a cross-reactive neutralizing epitope. So we went back to the serum from individuals infected with HIV-1 and HIV-2 and tried to look if they have P3 antibodies and if they neutralize HIV. And in fact, some of these guys neutralize both viruses. So now we are trying to purify anti-P3 antibodies and then see if, in fact, they have a neutralizing activity. And we think that this P3 that initially was seen as a good drug now can be seen as a potential vaccine candidate. The second story is this. We recently have been studying spirobeta-lactams produced in Coimbra by our collaborator, Teresa Pinho-Mel. And we found, amazingly, that these compounds that have a beta-lactamic core have a very potent activity against HIV, but also against Plasmodium and also against influenza virus. And these are the IC50 values, both for HIV-1 and HIV-2, so it's a nanomolar activity. For malaria, it's a micromolar activity, and it acts very well both at the liver and blood stages. We have modified this molecule many times, produced many derivatives, and found that many of these derivatives also work very well against HIV-1 and 2, and also against Plasmodium. They are very stable, they don't have any antibacterial or antifungal activity, and very small or no cellular and rat toxicity. We have tried to define the mechanism of action of these compounds using time of addition experiments. As you can see in this minute graphic, it works during all phases of the HIV life cycle. It doesn't have any protease inhibitor activity. We cannot select drug-resistant strains with against these compounds. It is synergic with other antiretroviral drugs. And so all of this points to a new and very complex mechanism of action, possibly working at the host cell and not at the virus. And so at this stage, we hadn't looked at the activity against any other virus besides HIV, so we decided to look at activity against other viruses, and with the collaboration of Helena Rebelo de Andrade from the INSA in Portugal, we decided to look at influenza, and we saw that it has also a very potent activity against influenza A and B, which is fantastic in our view. Perhaps we now can redirect this compound not for HIV but to treat lower respiratory infections, which are also a major problem, as you can see here. And so the idea is now to try to look at the activity of this compound against SARS-CoV-2, of course, and also against the respiratory syncytial virus. And perhaps this will be a drug in the future for lower respiratory infections and other virus infections. And so thank you very much. All this work, of course, was funded by Fundação para a Ciência e Tecnologia, Portugal.
Thank you very much. It was a very interesting talk. Does anyone have got questions? I have a couple, actually. One of them is in the orders. I mean, you use a really small amount. Do you know, in terms of toxicity, whether it elicits any toxicity with time, or whether it's safe for usage in animal models?
Yeah, we tried. We made some tests in Wistar rats with higher amounts, of course, and it was an acute toxicity assay, so small-term assay, and no toxicity was observed. We looked at toxicity at the liver level, at the kidney level, at all levels, and no toxicity was apparent. So we think that it's not very toxic. However, we don't know. The major problem in this type of compounds is the allergenicity, the hypersensitivity reactions that it may generate. In the rats, we haven't seen anything, but this is a problem in humans because many humans have a problem of hypersensitivity to beta-lactamic drugs. So we have to look at that more deeply.
And the idea is to be more towards a vaccine than a treatment itself, right?
We are betting more on the treatment.
But betting more into, do you know the time frame that in which you have to administer the drug for each of the individual pathogens that you have tested thus far?
Well, we only know in vitro, not in animal models.
Okay.
Except this macrobicides study that I have shown you, but it's external, it's a gel that we apply in the vagina of these mice.
But best of luck. It's very interesting, thank you.
Okay.
Any other questions? I ask again. Okay, then we can move on to the next speaker. And the next speaker is António Roldão from IBET, and we'll be talking about the advances in the bioprocess and analytical methods to accelerate the production of vaccines. And the floor is yours. Welcome.
Thank you very much. So I would like to start by acknowledging, of course, the organizing committee for giving the opportunity to present the work that we have been doing at IBET on the advances in bioprocessing and analytics to accelerate vaccine production. So I normally start by presenting this slide, which kind of summarizes the number of infectious diseases or outbreaks that has happened in the 21st century, so that you have an idea of that in just the past 20 years we have witnessed around, of course, 20 infectious disease outbreaks, starting from the SARS, which is CoV-1 in the early 2000s, to today with COVID-19. And it is, of course, expected with increasing mobility of humans, increasing consumption of animals, pets, as well as climate changes. The number of outbreaks that we have witnessed in the recent decades would increase and continue along, in the next years. So it's fundamental, the development or the tendency to generate bioprocesses for developing vaccines and innovative vaccines. But of course, the process of vaccine manufacturing is quite complex, and this is a graph that outlines the complexity that we have in the vaccine production platform. So each one of these phases are interlinked, and if you miss one of them, or there is an interruption, you have, of course, an impact on the public health and vaccination programs. We at IBET have been focusing on 5 of these steps, which are here highlighted in blue. and with the objective to accelerate the development of innovative and new vaccines. The way we have approached the complexity of vaccine manufacturing is from an holistic approach. So we like to integrate different technologies. So here we have integration of biology, with direct evolution, for example, upstream and downstream processing on the technology. We have the formulation, and we also have the analytics. So without this interlinked approach, then it's impossible to resolve or to solve our issues and to do with the complexity of vaccine manufacturing. In the next 4 slides, I will briefly mention four of these steps that are here highlighted. So I'll start with the biology component. So in the biology component, we have vast experience in optimizing the bioprocess. And in this particular case, what we wanted was to optimize our host cell that we normally use to produce our vaccine candidates, which is, in this case, influenza AJ virus-like particles, as a vaccine candidate against influenza virus, of course. And here, the idea was that we took the direct evolution concept, which is normally used for microorganisms, which is used, of course, to modulate the host phenotype by submitting it to typical temperatures or pHs or culture conditions, with the objective of improving a process. And in this particular case, what we did was to take our insect cells that we use to produce these influenza AJ virus-like particles and submit them to culture temperatures that are lower than the standard, in this case, around 22 degrees instead of the 27 degrees. And this took around 3 months that we did. And at the end of the day, of course, cut the long story short, we were able to increase titers by 7 to 8-fold compared to our standards, and also compared to the gold standard for the production of these virus-like particles, which is the insect cell baculovirus expression system. So at the end of the day, actually, we were able to improve the production in 2 different systems via this concept of direct evolution. So if we move on to the purification process, to the component of the purification component. So IBAT has, again, vast experience in designing and improving, for example, membrane cartridges or chromatographic matrices, and also continuous and multi-column chromatography. In this particular case that I mentioned here briefly, we wanted to move from a chromatographic-based process, which is sometimes costly and takes significant amount of time, to a membrane-based process that is normally based on disposable systems, for example, with the objective of reducing not only the time to purify our product, but also to increase the quality of the product at the end of the day. And again, to cut the long story short, the idea here is that our results show that we were able to recover from an average of 40 to 80% our influenza virus-like particles using membrane processes. But most importantly, we were able to collect at the end of the day influenza AJ virus-like particles, which are similar to what you have in nature for the influenza virus. So again, with this example, we demonstrate that membrane-based processes and purification processes for influenza AJ VLPs was successfully implemented. In the next component that I mentioned briefly at the beginning, we have the formulation side. So in this area, we have recently looked or explored the use of NADs, which is natural tactic deep systems, which are, for those who don't know, are a mixture of 2 or more components that by themselves are either solid or liquid, but when mixed together are liquid at room temperature and are normally used as, for example, cryoprotectants or stabilizers. In this particular case, we went to evaluate whether a specific mixture of trehalose and glycerol were able to improve the stability of our influenza AJ VLPs upon storage. And again, to cut the long story short, the idea here and the results show that we could not only reduce the degradation of AJ, which is the principal component of our influenza virus-like particles, by using a mixture of trehalose and glycerol that is around 90%, which is decreasing the KD from of course, the standard 0.26 to 0.06. but also we were able to maintain the integrity of our virus-like particles upon thermal stress, as you can see by looking at here at these graphs with a sharp peak for this mixture of T-glide 90%. So again, using natural athetic deep systems, we could improve the stability of influenza VLPs, and of course, those may have an impact on vaccine cold chain, not only for our VLPs, but also for many other vaccines. The final example that I have is on the analytical method side, and here, which we have, again, vast experience in designing and implementing these analytical methods, not only for product characterization, but also for process monitoring and control. And the idea here was to move from the traditional, for those who know influenza AJ, and the method that we normally use to quantify them, from the traditional hemagglutination assay that requires red blood cells, that doesn't allow you to follow the process from the beginning, i.e. from the production to the purification part, and sometimes, of course, are dependent on the type of red blood cells that you get. So the idea here is to move from this type of more traditional ones that are still used, of course, to a more robust one, which is using, in this case, biolayer interferometry. And with this technique, we're not only able to, characterize or estimate the concentration of HA in different, let's say, subtypes or valencies, subtypes H1, H3, Bs, or even Ns, but also for different valencies. In here, it's difficult to see, but from monovalent ones, which is just containing one AJ, to multivalent, either having 3 different AJs or 5 different AJs. So with the complexity with this system, we could tackle it. But also, we could also quantify or estimate AJ in bulk samples, I mean, from the production to the purification, which was really critical for us to design an improved and universal process for purification of our VLPs. So at the end of the day, with this technique, we could not only assist upstream, but also downstream monitoring and optimization. I would just like to have 3 more slides just to highlight not only our competencies here at IBET, but also, of course, our collaborations and our collaborations with other institutions, but also with GenIBET. So the first one is this slide that demonstrates the type of portfolio that we have generated along these 30 years of existence of IBET. So we have been focusing on human vaccines, from the beginning with HIV and rotavirus to more recent with SARS-CoV-2. But we also have been working on veterinary vaccines, and the most recent one being a vaccine against rabbit hemorrhagic disease virus, which together, of course, with our partner INIAV at the Agrotrec campus in Oeiras. And more recently, we also have been engaged in the consortium serology for COVID, which, of course, involves not only IBET, but 4 different institutions that are present here in this room, IGC, ITQB, IMM, and also SEDOC. But the slide here is to demonstrate that knowledge that we have acquired along these 30 years of existence of IBET was really critical, so that we could produce our proteins in a very short time that could be delivered to our partners to establish and to implement the method and to characterize the proteins as well. And I would like to finalize the presentation with this slide, which kind of, again, gives an idea of where we stand in terms of bioprocess development. So if you consider preclinical, clinical trials and licensing, so IBET works not only from the preclinical side, which is starting with small-scale production to pilot-scale production, but also then transferring our process to our partner GenIBET, which produces GMP lots for vaccines and for phase 1 and phase 2 clinical trials. We have also, should not forget that, of course, what we produce here at GMP needs to be released, needs to be certified, and that's why we use our analytical service units, which is GMP certified, that can, of course, release the lots for the clinical testing. And with that, I would like to finalize. Of course, this is just part of the people that was involved in all these projects, and of course, our funding. And thanks very much for your attention.
Absolutely great to see so much invention done in Portugal. Does anyone have questions? I would like to ask you whether within all these vaccines and the clinical trials that have been performed, whether you already have something in the market that is being used by all of us and we don't know?
So normally, I mean, IBET works more on the preclinical development. So essentially, we develop the processes to produce either a vaccine, it can be a protein, it can be a viral vector, it can be anything, you know. And sometimes, of course, the process is killed, you know, after phase 1 or phase 2 clinical trials. Other processes can go a bit further. What I know from our portfolio is that most of these vaccines are then, the process are then transferred to companies that, of course, buy or at least, you know, we transfer the knowledge or the technology to them, and then they start producing, of course, their own product. From my knowledge on the process, we have a couple of these products on the market that is being used, at least one for the veterinary vaccines, a vaccine against leishmaniasis, I guess. For the human vaccine, I'm not sure whether we have some in the market that we could, of course, tell.
And now a provocative question. Within the new platforms with mRNA technologies, how do they integrate your own platforms, and how would they be able to speed up this so lengthy process of starting to produce a vaccine until it's out there in the market?
I mean, we can, at the moment, we don't have processes with mRNA. Of course, Genabet, our partner that I just mentioned, has produced, you know, significant around these 15 lots of mRNA vaccines, not for SARS-CoV-2, for other indications, with a partner which was Moderna at that time. This was in the last 5 years or so. This is on the mRNA side, the knowledge that we have. At IBET, on the preclinical development, we never work with it. It's something that it's pretty much accessible or easy to do in our hands, taking our expertise. But I would like to say that, you know, I don't think it's the process of producing a vaccine of either mRNA or a protein or a viral vector is the main limitation. If you consider the time that you need to, from, you know, demonstrating that either the protein or the mRNA or the viral vector works until you have actually proved. This is exceptional case for SARS-CoV-2 because we are in epidemics, right? Because if you are not in epidemics, then this takes easily 10 to 15 years. And it's not because of the time that is needed to produce the vaccine. It's actually to guarantee the safety, to guarantee that they are not toxic, to guarantee that they work, that they provoke any side effects. So most of the time is not on, it's more on the regulatory and also on the safety side rather than actually the production side. So this to say that the technology producing mRNA vaccines, it's not, you know, that much complex as it is not for the other vaccines. And it can be done in a very short time frame, for sure.
Thank you very much.
You're welcome.
Okay, our next speaker is Henrique Silveira from the Institute of Hygiene and Tropical Medicine, and he will be speaking about the creation of mosquitoes that transmit malaria and that have been cultivated without using blood. And the floor is yours. Thank you.
Okay, good afternoon to everyone. First of all, I would like to thank the opportunity to presenting part of our work we are developing at the Instituto de Higiene e Ciência Tropical na Universidade Nova de Lisboa. And today I'm going to talk on raising mosquitoes for malaria research and control without the use of fresh blood. Just a quick general idea of the problem. Well, malaria in 2019 killed 409,000 people, and there are 229 million new cases of malaria. And the problem is this is mainly on sub-Saharan Africa and mainly on children under 5. So malaria, the parasite that causes malaria has a complex life cycle that needs a human and female mosquitoes from the Anopheles genus. So why are we interested in mosquitoes? It's because we want to block transmission. And why we want to do that? Because by blocking transmission or blocking the cycle in the mosquito, we would stop malaria. To do that, we need innovative control measures, and the majority of these innovative control measures available at the moment need mass rearing of mosquitoes, namely drug and vaccine clinical trials, release of massive numbers of mosquitoes into the wild, either sterile male mosquitoes or transgenic mosquitoes, and to do that we need high amounts of blood. This has ethical and financial costs. So, and at the moment, the stable production of malaria mosquitoes depends on fresh blood. So, and how do we do that, us and everyone else working with mosquitoes? We need human blood, slaughterhouse blood, or sedated or restrained live animals. This obviously it's expensive, it has safety issues, it has ethical issues, it needs qualified personnel and animal houses facilities. So to overcome this problem, we developed a diet that doesn't have fresh blood, and we tested in mosquitoes. And what we found out is that our diet performs very well and almost similar as blood what concerns fecundity and fertility, and also in the majority of the fitness parameters that we have tried there on the left. And why this diet is better? Because you can have high quality mosquitoes, you can reduce operational costs, you don't need blood at all. So where are we at the moment? We have published the diet, we have an international patent on the diet, and we've been on some entrepreneurship of the idea. But the most important, what keeps us going, which is we are very excited about it, is because we have an insectary running for almost 9 months, exclusively on diet that no blood has get into this insectary. And mosquitoes are alive and kicking, which is a very good sign for us. We are also developing further this diet. We were recently awarded a project to develop a network with 3 African countries, Burkina Faso, Tanzania, and Mozambique. And basically, we want to make sure that our diet is not working only on our mosquitoes in our insectary, but can be used widely. And also, we want to make sure that these mosquitoes are not selected and they still can transmit malaria, can be used on malaria research and control. And of course, we want to have a network of insectaries that work on and develop transmission blocking tools. We also are doing some microbiome and genome alterations that might occur with this diet. We are doing that with this S3 from EU, Infravac 2. And we're also trying to apply, to have this diet to be more universal and applying to other vectors of other diseases like Aedes aegypti, which is a vector from dengue, yellow fever, chikungunya, and so on. And we are doing that with the World Mosquito Program, with the group in Brazil. And we also have some ongoing activities of trying to get some funding with the Walter Reed Army Institute of Research and the nuclear, and I always get confounded with the name, but the SFO institution that work with mosquitoes. At the moment, we are looking for commercial partners because we are scientists. We don't know anything about entrepreneurship. Well, now I know a little bit because we've been to these workshops and so on. And we need a partner that can add commercial value to the idea and improve our existing business plan, and above all, to produce the diet in large scale industrially, and to finance the go-to-market or go-to-market strategy. We expect to have a diet, a universal diet that could be marketed in 2024. So this wouldn't be possible without our team, and it's me and João Marques, and originally we did the first essays with João Cardoso, Deborah Power, and Ruth Felix from CCMAR, Universidade do Algarve. And why do we keep doing this, and why do you think we are doing this is sort of important? If you look, how malaria was in the beginning of 2000, the 20th century, it was really a global disease, and it was all over the place. And it has been steadily shrinking, okay, till it gets to this, the actual situation when it's distributed mainly on tropical and subtropical, and the aim is to reduce it further and further till 2040 and get eradicated, or at least eliminated in most places. So we like to think that our work somehow is contributing to this global aim of reducing malaria and improving world health. And finally, all of this is not possible without financing, and this original idea was financed by Bill and Melinda Gates Foundation, and we have all sorts of financing. The last one from Gulbenkian FCT. I would like to thank especially our R&D Center, Global Health and Tropical Medicine, and the Nova Impact. That's helped us a lot and support us in trying to put this scientific idea into an enterprise or something that could be of some commercial value. Well, contact us if you want to know more about this diet. And if you would like to invest in this unexploited growing market of mosquito diets, please contact us. That's our email address. And thank you very much. Obrigado for your attention.
Okay, now you know to contact if you're working with malaria and need mosquitoes. So, are there questions from the audience?
Well, of course, we all know that we are what we eat.
Yeah.
So, are these mosquitoes indistinguishable from the ones that feed on fresh blood? First question. And second is, are the raw materials that you have not mentioned, but are they widely available in poor countries, or is it a problem of having access to the raw materials to make up the diet?
Okay. The first one, morphologically, they are not, you can't distinguish them. If you put them together, they will be exactly the same thing. We've done some measuring of the wings, which is normally a standard measure for fitness of the mosquitoes. And there's no great alteration, or they are similar. They vary a little bit between blood fed and diet fed. And if the diet is available to poor countries, that's what you're asking. We don't use fancy, it's just nutritious elements. The idea is not to profit out of it, it's just to make it available. If you're asking me if it is, if you can compete with certain blood supplies, some of them not. In our case, that we depend on animal house and small rodents. Yes, it's very competitive because we have to do it in our animal house. But for instance, for insectaries that rely on slaughterhouse blood, then we are not competitive at all. Again, if you think on blood, it has a very short shelf life. It needs to be refrigerated all the time. it is you have a lot of variation. I don't know if anyone here worked on mosquitoes, but it's a nightmare when it comes to statistics because it's variation all over the place. Whatever you do, it doesn't matter if the population is in your insectary for 10 years, you still have lots of variation. So with these diets, we will expect to have to even this variation. Yep.
More questions from the audience?
Do they transport plasmodium?
Yes, they can. In fact, if you infect them experimentally, they can transmit Plasmodium bergii, and they can be infected by Plasmodium falciparum. Although this is not fully ready for publication, but we've done it in our lab, and we've done it in Burkina Faso and Tanzania, and yes, they can be still infected with Plasmodium.
One of the strategies of controlling viral vectors at the moment is by releasing mosquitoes with Wolbachia.
Yes.
Do you know whether the mosquitoes fed on your diet and also containing Wolbachia, whether they would have exactly the same role?
Yes, we did some, I don't know if you remember, but we have some experiments with the Aedes aegypti, with this group in Brazil, and they were particularly interested because they are releasing mosquitoes with Wolbachia, and we did some experiments. They are preliminary, but we did some, there was some reduction on fecundity, for instance, if I'm not wrong, but on Wolbachia infection, there was no difference whatsoever. Okay.
Thank you very much. It's very nice. Thank you.
Okay.
So,
well, thank you all.
The next speaker of today is Sonia Dias from, and now I'll switch to Portuguese because she's going to do her presentation in Portuguese, from Centro de Investigação Integrada em Saúde da Escola Nacional de Saúde Pública, que nos vai falar do barómetro Covid-19, opinião social, conhecer, decidir, agir, os portugueses a Covid-19 e as respostas do Serviço Nacional de Saúde. Obrigada.
Okay, so first of all, good afternoon. Sorry, I prepared my presentation in Portuguese. So if you, any of you do not understand Portuguese, please talk with me after the session. Portanto, boa tarde. Vou então falar sobre o projeto Barómetro Covid Opinião Social, um projeto financiado pela FCT. Este projeto surge face à ausência de informação oficial sobre como é que os cidadãos estavam a viver a pandemia e também compreender os seus efeitos no seu dia-a-dia. E, portanto, a Escola Nacional de Saúde Pública da Universidade Nova de Lisboa lançou o projeto de Barómetro Covid-19 Opinião Social, com o principal objetivo de acompanhar a evolução e o impacto da pandemia na perspectiva dos cidadãos e perceber um bocadinho os efeitos na sua vida e no seu cotidiano. Portanto, este projeto pretendeu monitorizar os efeitos da evolução da pandemia e com isso, e para isso recolhemos informação sobre percepções, comportamentos, mas também alguns indicadores socioeconómicos, indicadores de saúde e também sobre os efeitos das medidas de combate à pandemia. Conseguimos com isto também identificar vulnerabilidades sociais e em saúde, resultantes do confinamento, e também analisar os impactos das medidas específicas implementadas pelas autoridades de saúde junto da população. Este projeto também procurou analisar de uma forma rápida, em tempo útil, a situação atual e identificar tendências ao longo das fases e da evolução da pandemia. E, por fim, também este projeto procurou translação da evidência em práticas efetivas através do delineamento de estratégias operacionalizáveis que contribuíssem para mitigar o impacto da pandemia, para melhorar a utilização dos serviços e também para reduzir as desigualdades. Em termos de desenho do projeto, uma primeira componente do projeto incidiu na produção e disseminação de evidência através de um inquérito online, que já irei falar de seguida. Uma segunda componente de translação da evidência, focada no acesso, em promover o acesso adequado e equitativo aos serviços e também à literacia em saúde, através da condução de painéis de peritos. Este projeto também veio potenciar a criação de redes de colaboração de investigação a diversos níveis. Por exemplo, com vários países, um deles Brasil, que neste momento, atualmente, está a decorrer o barómetro, que é o termómetro social Brasil, mas também permitiu desenvolver parcerias noutras áreas, ao nível da saúde mental, da violência, mas também de outras doenças. Relativamente à produção e disseminação da evidência, nós desenvolvemos um inquérito de preenchimento online que foi iniciado em março de 2020, no início da pandemia, dirigido à população em geral. Tratou-se de um inquérito dinâmico, portanto, determinados blocos de perguntas eram regularmente adicionados ou retirados de acordo com as necessidades e a própria evolução da pandemia. Obtivemos mais de 190 mil respostas, com uma recolha de dados regular, quinzenal, de mais de 175 mil respondentes. Este projeto permitiu, desta forma, também uma componente de estudo longitudinal e, portanto, permitiu uma análise de tendências ao longo do tempo. As principais dimensões que pudemos monitorizar passaram pelas medidas, pela adoção das diferentes medidas de proteção individual, como o uso da máscara, o distanciamento, a higienização das mãos, mas também a percepção individual de risco de infecção e de gravidade da doença. Também as questões da confiança do indivíduo na capacidade de resposta, quer das autoridades governativas, mas também dos serviços de saúde, o acesso aos cuidados de saúde ao nível das consultas, dos serviços de urgência, tratamentos, mas também ao nível dos cuidados preventivos, exames e também acesso à vacinação. Obtivemos também informação sobre indicadores de saúde mental e bem-estar e também sobre desigualdades sociais e em saúde. Nós, com este projeto, sabemos que não conseguimos alcançar determinados grupos mais vulneráveis e, por isso, tomámos a iniciativa de desenvolver uma segunda componente, um estudo focado em populações particularmente vulneráveis, como são as populações migrantes. E, portanto, nesse estudo, nós aplicámos um inquérito por questionário a um total de 1.091 migrantes de várias origens, e residem em Portugal há menos de 10 anos. Este questionário foi aplicado por entrevista por inquiridores bilíngues e o instrumento foi disponibilizado em diversas línguas. Estas são as principais dimensões do instrumento que foram exploradas. Por um lado, a caracterização sociodemográfica e o contexto de migração, mas também a percepção de risco em relação à infecção e a adoção de medidas de proteção individual e depois as questões do impacto da pandemia, quer nas condições de vida, em termos de situação financeira, situação laboral, mas também ao nível da condição de saúde, incluindo a saúde mental, e também em termos de acesso aos cuidados de saúde durante o período da pandemia. Depois, numa perspectiva de translação da evidência, nós procuramos que esta produção, que esta evidência produzida pudesse, de alguma forma, contribuir para informar quer autoridades oficiais, com conhecimento para fundamentação estratégica, para suporte de decisões, mas quer também serviços de saúde, por exemplo, com a recolha de informação sobre a adesão, a confiança, a percepção de segurança ou a própria aceitação de estratégias que foram sendo adotadas, como, por exemplo, a telemedicina. Pretendemos também informar, de alguma forma, os cidadãos em geral, identificando necessidades em saúde e também vulnerabilidades. Numa fase Com base nesta evidência, numa fase posterior, fomos então, procedemos à consulta de painéis de peritos, e dessa consulta resultaram recomendações concretas de estratégias de ação que pudessem contribuir de forma genérica para mitigar os impactos da pandemia. Não querendo alongar muito também, devido à limitação de tempo, só aqui mostrar-vos que estes foram as principais áreas de recomendações que surgiram dos dois painéis. Portanto, o painel de peritos focado nas questões exploradas no barómetro Covid-19. As recomendações abrangeram 3 áreas específicas: as questões da reaproximação dos cidadãos aos serviços de saúde, o incentivo à vacinação e a promoção da saúde, incluindo saúde mental e estilos de vida saudáveis. Relativamente ao painel de peritos que foi realizado, focado nas questões dos migrantes e pandemia, estas recomendações abrangeram 5 áreas concretas, com vista a mitigar os efeitos da pandemia nos determinantes sociais da saúde, reduzir as barreiras no acesso aos cuidados, promover o acesso e a utilização da informação em saúde, atenuar o impacto da pandemia na saúde e, em especial, na saúde mental, e também combater o estigma e discriminação. Portanto, no âmbito deste projeto, foram desenvolvidas várias iniciativas de disseminação e de comunicação dos resultados que fomos obtendo. De forma genérica, a disseminação, os resultados permitiram prestar ou dar informação pública, tornando pública, para informar, por exemplo, decisões político-estratégicas. Também foi possível desenvolver um dashboard dinâmico com gráficos interativos. É um dashboard que está acessível, é de acesso aberto, está acessível no site da Escola Nacional de Saúde Pública. Também houve uma disseminação estratégica nos mídia. Produzimos relatórios públicos com os principais, que sintetizassem os principais resultados dos projetos e direcionados quer para as entidades, quer para os serviços de saúde e profissionais e também para os cidadãos em geral. Em linha com os relatórios, também foram produzidas infografias para uma comunicação mais simples também à população em geral. Em termos de divulgação científica, temos já publicado alguns artigos com resultados deste projeto, também algumas apresentações em eventos e também já este projeto e esta informação já está a servir também de base para algumas teses e trabalhos académicos. Deixo-vos aqui, isto são exemplos, são as capas dos relatórios públicos que estarão, os do barómetro estarão em breve disponíveis, o das populações migrantes já está disponível no site da escola, para quem tiver interesse em conhecer um pouco melhor. E obrigada.
As questões em português ou em inglês, Sónia?
Pode ser em inglês.
Okay. Anyone has got questions for Sonia?
Sorry, just to... o meu nome é Ana Gama, eu não sou Sónia.
Ah, pois é, veio a substituir, não é?
Exato.
Peço imensa desculpa. Eu tenho uma questão. I do have a question regarding, for example, children. How children that live in complex families, have you done anything in order to understand, or have you looked onto the problem on how to understand how confinement or how studying from home has actually the type of impact it has had on the development and in the conditions of these children?
Indeed, that is one of the most vulnerable subgroups, but our study did not reach that group. It was only adults. But from the data and the findings, we foresee that there is also impact on their children. We saw a tremendous impact on the economic impact on lives of many people, and also on mental health. So we anticipate that this also has affected the household living. Yeah.
And are there any guidelines for whether this ever happens to us again, God forbid? Are there any guidelines on how to be able to approach this type of situation in a better way that deals with mental health and other issues as well?
I think that is the $1 million question. Because indeed, I think that is the great challenge we have to face, is how this pandemic brought to light inequalities, existing inequalities, and aggravated even more the inequalities. So there are a lot of work to do. I heard someone telling that we have several new pandemics: economic pandemic, the mental health pandemic, domestic violence pandemic, just coming just as a result of COVID. So that is really a challenge for all of us, and in terms of, and politically, to how to best address and to mitigate the effects, prevent, and also to support.
There's a question from António. Henrique, sorry. Sorry.
Well, no problem. Just joking. On your migrant study, do migrants have similar access to health system and vaccines concerning COVID?
Well, indeed, in our sample, we were able to reach very different migrants. Most of them were in regular situation, but about 10% were undocumented. And that is, from the beginning, a factor that influences access to health services. We found that, indeed, those who were socially more vulnerable, so those undocumented with less income, with lower income, non-employed, were the ones who reported more difficulties in access during the pandemic.
Any other questions? If not, we just have to thank again Anna for coming, and we welcome the next speaker, who is Richard Inman, from the Centro de Investigação em Psicologia para o Desenvolvimento da Universidade de Lusíada. Thank you, and the floor is yours.
There we go. Hi, everyone. Thank you very much for receiving me, and the talks have all been very interesting. I want to start just quickly by saying the topic of my talk is somewhat different from what we've had before. It's not on infectious diseases, but I'll try my best to see if I can link it in. And I'm going to start with the question, how involved, how engaged are you with sustainable development? So, it is undeniable that to achieve sustainable development goals, that we need as many people as possible to be able to modify their attitudes and their behaviors towards sustainable development. To try and link that in, we've been talking about vaccinations. On the one hand, it's great to be able to develop amazing vaccines, but if we can't convince people that they're useful, and if we can't convince people to go and get their COVID-19 vaccination, then we've still got a serious problem on our hands. Because we've got this need to understand behaviors and attitudes, there is a chance for the behavioral scientists and psychologists like myself. But this leaves a question. When we're trying to understand human behaviors and human attitudes towards sustainable development, at what level should we be addressing our research? And at what level, when we understand it, should we be addressing our interventions? For example, should we be trying to understand how to promote attitudes and behaviors for the individual sustainable development goals? Or perhaps we should try and aim a little higher and try and convince people to modify and empower them to behave towards the 3 major pillars of sustainable development. Or perhaps something higher level. Oh, I don't have a pointer. No problem. At CIPT, we've argued that an effective framework for promoting sustainable development should address how humans relate to all of the sustainable development goals as a holistic issue, rather than being specialized and domain-specific. And the reason for this is that all of the domains of sustainable development have something in common, which is that people relate to them through the same underlying psychological processes. That is, we have subjective experiences of them, and we identify with them, and we connect with them. And these result from an interaction between our emotions, our cognitions, our thoughts, our behaviors, and the way that the context, the environment, interacts on us. Therefore, any effective, parsimonious approach to promoting or modifying behavior change, or empowering people to change their attitudes and behaviors, should fully capture how people's thoughts, feelings, and actions interact to shape their subjective experiences. Having said this, we noted that there is a gap in the literature in the sense that, okay, behavioral scientists have been interested in sustainable development issues, but often they have a narrow approach. For example, we have numerous constructs which have been designed specifically to address how we relate to environmental issues, nature relatedness, nature identity, environmental identity, and this sort of thing. And because of this, many of the associated measurements, questionnaires, also have a narrow focus. The problem is that when we have narrow approaches and theoretical constructs which are specific to a certain type or area of sustainable development, this means that we have numerous measures, we have numerous approaches, numerous constructs which are hard to integrate, and then we have very specialized and independent lines of research and intervention which are not transferable. So, at CIPT, we decided to export the construct of engagement, or in Portuguese, Desenvolvimento, from educational psychology. Engagement is ideal for this because it has a numerous set of qualities. It is itself a multidimensional construct that captures the sense of identification and connection with a topic. It is multidimensional in the sense that it captures our emotional experiences, our cognitions, and our behaviors towards something. It is robustly validated in educational psychology in the context of engagement with school. Critically, engagement as a construct is particularly predictive of positive outcomes. In terms of education, of course, this is academic performance, school attendance, but applied to sustainable development, outcomes will be the way that will be sustainable development goal outcomes. More critically, engagement is highly malleable in the sense that it interacts and is influenced by the environmental factors, such as policy, intervention, our social support, and this type of thing. In other words, engagement may be a critical locus for intervention applied to sustainable development. So at CIPT, as part of a FCT finance project, we developed a questionnaire to measure people's engagement with sustainable issues as a holistic topic rather than specific goals. And this is called the Engagement Disengagement with Sustainable Development Inventory, the EDIDSI. This questionnaire has 6 dimensions. Firstly, we have behavioral, cognitive, and emotional dimensions of engagement with sustainable development. Critically, we also have behavioral, cognitive, and emotional dimensions of disengagement with sustainable development. This is important. In psychology, we consider the mental well-being as not being equivalent to the absence of psychopathology. In other words, being healthy is not the same as the absence of illness. And in the same sense, being disengaged with a topic is not the same quite, or not the polar opposite of being engaged, although they are negatively correlated. So, this structure we have found with numerous samples of large sample size in several different contexts, as I will show you, has been validated using exploratory and confirmatory factor analysis. Having designed this questionnaire and applied it to numerous samples, I'm just going to summarize some psychometric evidence. So, first thing, this questionnaire has what we call measurement invariance. We applied it, first of all, in a sample of Portuguese adults and a sample of British adults, university students at the University of Essex in the UK, and the same factorial structure emerges. In other words, the items measure the same things, and engagement with sustainable development is the same thing for British and for Portuguese adults. We also performed the same analysis for samples of Portuguese adolescents. There's a sample of about 3,000, and Portuguese adults, university students again. And once again, we find that the construct means the same thing. This is exciting because it means it has the potential to be used for international research, although would require more translations and further validation in this sense. Second finding, in order to be convinced of the robustness and the validity of your construct and your questionnaire, you need to be able to prove that it has validity. In this case, this slide shows convergent validity. Validity is it measures what you purport it measures. And you can do this by performing simple correlations with theoretically related constructs. As I can show you on the top, individuals who score high in engagement with sustainable development also tend to score high in environmental action. And the reverse, those who score higher in disengagement tend to have lower scores in environmental action. And we have got similar results for biospheric values and altruistic values. Those who are engaged are concerned for the environment and they're concerned for others, while those who are disengaged tend to be the opposite. In other words, it seems that the construct measures what we purport it to measure. In a similar analysis with Portuguese adults, this is a regression controlling for age and gender. We find that engagement is predictive of pro-sustainability behaviors. This is not longitudinal, by the way. I say predictive in predictive of variance sense, and is also predictive of pro-environment behavior and motivation. Finally, as I said before, engagement and disengagement theoretically are not polar opposites. And to test this, we got the individual scores of engagement and individual scores of disengagement, and we performed a latent profile analysis to find emergent subgroups. So we've got the two axes here. One is an engagement axis, another is a disengagement axis. And most clearly, we can see that there are 6 groups which cross the axis, which suggests that, well, yeah, by and large, people who score high in engagement score low in disengagement and vice versa. But proving that they're not entirely polar opposites on the same continuum, you have other groups of adolescents, in this case, that score high in disengagement and high in engagement. In other words, they are a bit mixed message. We then did a quick analysis to find out how these different profiles were related to some other variables. Just to quickly summarize, basic psychological needs, so when individuals feel that they are competent, autonomous, and related, should help individuals be engaged. And we found that those who are engaged and not disengaged tend to have higher basic psychological needs. In other words, when we get the psychological nutrients that we need for well-being and psychological functioning, we tend to be more engaged. I'll leave that there and move to the conclusions, which is we've developed an instrument for measuring engagement with sustainable development, and thus far, at the beginning of our set of analysis, we've shown that it seems to be a valid and reliable instrument. We now have a completion of a longitudinal 4-year study in which we will be analyzing how, one, it changes over time in adolescence. I say fortunately, this spans the COVID pandemic. So this will be interesting for us because it allows us to see how that actually changes as a function of the emergence of the pandemic. This study involves about 100 schools, and we measure school-level characteristics, which means we'll be doing a multi-level analysis to understand what features of schools help to promote and maintain engagement with sustainability in students over time. The fact that we've translated this into English and had that validated over there suggests that, well, this may be the beginning of something international, and indeed we are in talks with some collaborators about that. Engagement fills an important gap in the literature as a construct that explains how individuals experience sustainable development as a holistic issue. And the instrument itself fills a gap for the same reason. And we believe it has the potential to be used in a whole range of contexts. For example, educational effectiveness of education for sustainable development, for evaluating the implementation of any interventions or policy. So, to conclude, thank you very much. I would like to thank the FCT, also my supervisor, Paulo Moreira, and other members of CIPT for their research. Thank you very much. Obrigado.
Thank you very much. It's an interesting topic. Do we have questions from the audience? I would like to ask you something, whether critical thinking would be a critical ingredient in order to activate engagement in whatever field that we could look for, and how do you think that this could be achieved?
Well, you say critical thinking, which immediately takes me to thinking about the cognitive aspect of engagement. So I did not specify, but cognitive engagement incorporates a variety of things, including, for example, motivation, but also adaptive, pro-sustainable thoughts. So critical thinking as a component, as a cognitive exercise, I certainly believe would be useful with promoting the cognitive aspect of engagement.
Thank you. We don't have time for more questions, but there's one from Anna.
Thank you very much. The presentation was very, very interesting. Do you foresee to test the instrument with an older population?
Older in what sense? In the sense of a geriatric?
Older than university students?
Yes. So the results I presented cover a numerous series of experiments. So I said that the British sample is essentially university students, but the Portuguese adult sample is not just related to university students. It incorporates university students, but it also includes older range, up to about the age of 80, I think. So we believe is fairly representative of the different age ranges. Welcome.
Thank you very much.
Thank you.
And now for the last speaker, who is Catarina Pimentel. She will be talking on behalf of Most Micro ITKB COVID-19 Scientific Task Force and about Most Micro ITKB emergency research tackling COVID-19 from basic science to applications. Thank you, Catarina, and the floor is yours.
Okay, so good afternoon. It's my great pleasure to be here today on behalf of the Scientific Task Force of Most Micro ITQB to give you an overview of the projects ongoing in UNIT within the scope of the pandemic. So I would like to thank the organizing committee for this opportunity. So first, let me tell you how it all started. So 1 year ago, during the first lockdown, all researchers of ITQB received an email from the ITQB's direction inviting them to attend a general online meeting where the role of ITQB's research and within the context of the pandemic would be discussed. So some of us decided to bring up ideas for discussion, and which actually ended up receiving an initial funding from Most Micro ITQB to be further developed.
Katarina, sorry.
I'm sorry.
You can remove the mask to make it easier to...
Okay, thank you, sorry for that. You got used to it, right? So, okay, so and I would say that this was actually the birth of our task force. The projects ongoing at Most Micro ITQB can be roughly divided into 4 categories. So the rational design of antivirals, SARS-CoV-2 proteins, oral vaccines, and diagnosis and surveillance. So the idea of the colleagues working in rational design of antivirals was to design antiviral proteins that bind to the spike protein of SARS-CoV-2 and block its interaction with the host receptor, which is ACE2. So, of course, this way, preventing the virus from entering the cells and replicating. So, and this slide is just to remind you that the spike protein is that sharp bump that protrudes from the surface of the virus, and the binding site to ACE2 receptor is also known as RBD. So RBD is a name that I'm going to evoke very often during this presentation. So there are 2 projects ongoing under this category. As you can imagine, they are deeply rooted in computational biology, and they use slightly different strategies. So the first one is led by Diana Loza, and it took advantage of the information on the structural it took advantage in the structural information on the interaction of ACE2 with the RBD. To produce chimeric proteins. So the idea of Diana and her team was to use a motif of ACE2, here in green, and then grafted it into a stable scaffold protein. So after the protein design, the corresponding genes or the corresponding sequences were cloned, and then the proteins were expressed and purified in E. coli. And of course then characterized to see whether they could indeed bind to the RBD. So from the initial 5 candidates, 2 are already ready to go for infection studies. So while I'll be more than glad to take any questions that you may have at the end of this presentation, I decided to include the contact details for each coordinator so that you can contact them if you want to know more about each project. So in the other project led by Manuel Melo and Isabela Abreu, instead of using dead chimeric proteins, they used ACE2 itself, and they also designed spike neutralizing antibodies. So the strategy here below, it's quite similar to the one I've shown you earlier, but perhaps the main difference is that instead of using E. coli cells, they express their proteins in the plant model Nicotiana bentamiana. So most of the projects fall into the next category, and actually this category includes 2 different types of projects. So those seeking to develop cost-effective platforms for antigen production that can be then used, for instance, in serological tests. Rita Branches is expressing RBD and spike protein in tobacco BY2 cell lines, whereas Ricardo Loura and Cantaria Paquete are doing the same, but using E. coli cells. So, and this category also includes more fundamental type of projects, so to speak, where at least 3 teams are studying relevant SARS-CoV-2 proteins. And this is the case of Célia Romão and her team, who, using biophysical and structural approaches, want to understand the molecular details underlying the interaction of NSP1, which is a viral protein, and the host proteins. Of course, the ultimate goal of this kind of projects is to devise new therapeutic approach. So another protein of interest is the nucleocapsid, or N protein for short. And so the N protein is very, very abundant, and it is very important, or it is crucial for the viral RNA packaging. So it's important for this process. However, the molecular mechanisms underlying the process are far from being well understood. And this is the question that Tiago Cordeiro and his team want to answer by using a combination of several methodologies. And the last project within this category is dedicated to the study of the 2 ribonucleases, so the NSP14 and the NSP15. So these ribonucleases are important for viral replication, and the team has already performed their biochemical characterization, and actually this has been very recently published. So, and also they performed an in silico screening for chemical compounds able to inhibit these proteins. And this was very interesting because they came up with 3 drugs that indeed, at least in vitro, are able to inhibit the activity of the 2 RNases. And most importantly, they are also able to prevent infection. So a patent is in progress. In the right panel, I'm only showing the results for one of the drugs, but they have obtained similar results for the 2 other drugs. So in the category of oral vaccines, there is one project aimed at using Bacillus subtilis spores as a display platform for oral vaccination. So the team came up with the idea of fusing the RBD of SARS-CoV-2 to an endogenous protein of Bacillus subtilis, which is natively expressed at the spore surface. And using this strategy, they were indeed able to display several or a large amount of RBD molecules at the surface of the spore, as you can see in the middle panel. And this image is an immunofluorescent image obtained with an antibody and anti-RBD. So, and now they use these spores to feed mice, and immunization studies are on the way. So, as for diagnosis and surveillance, as you, some of you probably know, ITQB is part of the Portuguese network for diagnostic of laboratories for COVID-19. So from Monday to Friday at ITQB, the public servants are screened by RT-PCR for a symptomatic carriage of SARS-CoV-2. And as you know, this is extremely important to avoid the COVID-19 outbreaks. So this is a partnership between the Oeiras municipality, the local health care centers, primary health care centers, and of course ITQB Nova. And as for June 2020, more than 10,000 tests have been performed, and most results are reported within 10 hours of sample collection, which is quite impressive. And this is, of course, only possible thanks to the great effort of Raquel Salião and their team, who set up the facility in record time. and the daily RT-PCR analyses are performed by a group of highly motivated volunteers whose names are listed in these slides. So we are also very interested in cheaper, faster, and simpler alternatives to RT-PCR testing, and therefore we set up an RT-LAMP-based assay. So as RT-PCR tests, the RT-LAMP also detects the viral RNA, but in a much more simple way. So it uses a completely different set of enzymes. And at ITQB, we have two different formats of the test. We have a fluorescent format and also a colorimetric format. But today I'm going only to talk about the colorimetric format. So, and the readout is, you can see it in the right panel. So it is dependent on pH, because during the amplification there is a drop in pH, and the positive test is yellow, whereas the negative test it's pink. So we also have different flavors of the colorimetric test. So it can be performed from nasopharyngeal swabs or from saliva samples. Both tests have a good specificity and sensitivity, which make this test good enough for surveillance purposes. And we have also set up a new colorimetric detection method, and the patent application was submitted last year. We produced all the key components for this test, I mean the enzymes and also the colorimetric buffer, which allowed us to set up an in-house made test. So if you want to know more about this, our preprint is available. I'm the coordinator of this project, and it results from a partnership between ITQB Nova, Centro de Investigação da Academia Militar, INIAV, and the Hospital das Forças Armadas. So, and here are the names behind, or the faces behind the names I showed you throughout this presentation. Claudio Soares is the coordinator of the task force, and on behalf of all of us, I would like to thank everyone outside ITQB involved in this work, all the participating institutions, and FCT for funding. And thank you very much for your attention.
Absolutely spectacular. Questions for Catarina? Eric?
Well, I've got a question, but this is very nice, actually. And what I would like to know is this task force, how is it managed? And it's mainly a group of different projects, or projects feed on each other to maximize results?
Well, we have, I mean, well, we have different groups of ITQB Nova, right, working in the same project, okay? And also, of course, as I mentioned, we have also other institutions also contributing to the projects. So, so every month we meet together to present the state of the art of the projects, okay? And we have really great discussions. And so this is how it works, actually.
Excellent, thank you.
Okay. One question that I have is on the lamp that you have, the RT lamp with saliva. What's the cost of each of the tests that are performed, and how easy would it be to perform, say, for example, in a school?
Well, it's really, really very inexpensive. So I would say that less than €2 per sample, okay? And this is why I really think that they are suitable for surveillance. So RT-LAMP as antigen tests, they are not as sensitive as qRT-PCR, right? But as they are cheaper than RT-PCR, then you can do it more often, right? You can do it every week, for instance. And that's why I think they are really suitable for this kind of things, like, you know, screening children every week in the schools, so, yeah.
And I also ask because, as you know, I'm very involved in doing that, so I disclose this immediately. Yes. So other questions from the audience? No more questions? So we just have to acknowledge everybody and all the speakers here in this room. This was really a spectacular set of talks, and I thank you all for being here up till so late that we started late and we also extended the delay. So thank you all.